In the last section of this module, we're going to focus on the other anaerobic pathway. And if you'll remember by anaerobic, what I mean is that this process does not require oxygen in order to produce ATP. The process here is glycolysis. Glycolysis is an anaerobic pathway used to transfer bond energy to turn glucose ultimately into ATP by rejoining the inorganic phosphate to ADP. It produces this ATP relatively rapidly, not near as fast as phosphocreatine system can create ATP, but faster than the aerobic systems, which we'll cover in the next module. So how does it do it? It takes glucose, which again is a 6-carbon sugar, or glycogen, which is again a polysaccharide filled of many molecules of glucose, and it takes these to form 2 molecules of pyruvate or lactate. This occurs in the sarcoplasm of the muscle cells, again not in the mitochondria, which is where the oxygen needs to take place. This is just in the middle of the cell. However, in order to do this, we need to couple the reaction, right? The idea that we must prime this by the addition of ATP. And again, this is coupling these endergonic and exergonic reactions. So if we look at the picture here to kind of review this slide, there is an energy investment phase, right? So our glucose comes into the cell. We will ultimately add 2 ATP to this glucose molecule. But at the end of the day, we'll get an energy generation phase of 4 ATP produced. We'll also produce 2 NADHs and 2 pyruvates or lactate. So again, if we look at the output, 1 glucose gives us 2 pyruvates, 2 ATP go in to give us a net of 2, a total of 4, and 2 NADs get converted to NADHs. So the first step in glycolysis is starting the glycolysis process with a molecule called glucose 6-phosphate. That phosphate obviously gives you the idea that that phosphate is going to need to be added to a glucose molecule, right? So in our cells, we have in our muscle cells, a limited amount of glycogen that's stored, and so we can use that, but most of our glucose is going to come from blood glucose. If glucose comes into the cell, you'll notice it comes in as blood glucose. The first thing that we have to do is convert glucose into glucose 6-phosphate. The way that we do that is by adding an ATP molecule onto glucose to make this glucose 6-phosphate. So you'll note that this is the first ATP energy investment step. Why is this step important? It's important because essentially glucose follows a concentration gradient. So the way it opens is they have transporters that just open a hole into the cell and then glucose comes in. If it remained as glucose, it could go in and out depending on the concentrations. So therefore, the importance of adding this phosphate group, it changes the structure slightly enough that it doesn't fit in a transport cell. And then, so therefore, the first step in phosphorylating glucose to get glucose 6-phosphate by adding this ATP effectively traps glucose in the cell, still in this usable form, to then proceed down glycolysis. However, in glycogen, when it is broken down by its enzyme, then it actually does not need to be phosphorylated. It already comes in the format of glucose 6-phosphate. So if we take a look at that, how that works then, if we look at this same slide that we had earlier of the energy investment phase, glucose comes in, we require 2 ATP. The difference is if glycogen is broken down by glycogenolysis, we already have a phosphate on the molecule, therefore we only require 1 ATP. The same steps are followed as previous with glycolysis. We still get a net of 4 ATP, 2 NADH produced, and 2 pyruvates or lactate. So therefore, if we're starting with only having to put in 1 extra ATP when starting with glycogen, you'll note now that our net production actually goes up. So our net production of ATP from glycolysis when we break down glycogen is 3 ATP as opposed to the 2 ATP that we would normally see in glycolysis, starting with glucose. So here's what glycolysis looks like, and I know all the students look at this and go, oh man, that's a lot of steps. What am I supposed to know here? So I'll kind of highlight the ideas of what we need to know and how this works. So again, as I mentioned in the other slide, you need to know that when glucose comes into the cell from blood glucose, it is immediately phosphorylated, right? So this is a 6-carbon molecule. You'll remember that glucose here is a 6-carbon molecule. So glucose is C6H12O6. So again, a 6-carbon molecule. When it comes in, we have 6 carbons. The first thing that we immediately do, I'm going to draw this as a ring here because that's ultimately how glucose looks, is we're going to add a phosphate group. So that is step number 1, is adding a phosphate group as soon as glucose comes in. The purpose of this is, again, as I mentioned, trapping glucose into the cell. It's also investing in ATP that we're going to get out of it later. You'll notice we'll just skip through these. You don't need to know this specifically. We know that makes glucose 6-phosphate. That gets converted to fructose 6-phosphate, bringing us to the next step in which you need to remember. That is fructose 6-phosphate then gets converted into fructose 1,6-bisphosphate. So what does that name tell us? Bis means 2. You can see the positions here, 1 and 6. So ultimately what we do in this step is we add a second phosphate group onto glucose. There's a very special specific enzyme here called PFK, or Phosphofructokinase. This is a really important enzyme because what we call it is the rate-limiting enzyme. What do I mean by rate-limiting enzyme? That means that the overall rate, so how much ATP, how much the process of glycolysis, how many times it churns through glucose molecules, is ultimately going to be determined by how fast phosphofructokinase activity goes. So for example, if some way phosphofructokinase is increased in activity, then all of glycolysis is going to work faster. Right? And the same is conversely true. So if we slow down PFK activity, then all of glycolysis is going to proceed slower. That's not the case for the other enzymes. So for example, this first enzyme is called hexokinase. You don't need to remember that. But we can increase hexokinase up or down, and it ultimately won't have much effect on the ultimate rate of glycolysis. That is geared only towards phosphofructokinase. From a physiology standpoint, this makes perfect sense, right? We don't need to regulate all kinds of enzymes in these steps. We don't need to remember every single enzyme and have some type of way to modulate each one of them. Simply put, the body picks one enzyme, an important enzyme, right? It's an energy investment state. We're wasting an ATP if we don't continue down the path. So if we can stop glycolysis here without investing in another ATP, right? You can ask, well, why don't we regulate hexokinase? Remember, one, if we don't phosphorylate it, then the glucose leaves and it's virtually worthless to us, right? So we're not wasting an ATP here. We're trapping glucose in the cell in this situation. Here, we need to regulate so that we're not wasting ATP and keeping it at its maximal number. So again, phosphofructokinase, really important. A rate-limiting enzyme. You need to know that what it does is adds a second phosphate group onto this, and it ultimately controls the rate at which glycolysis proceeds. If it increases, then we run through this pathway much, much faster. If it decreases, then the overall rate of production from ATP from this process will, of course, then decline. So again, we enter the cell, phosphorylated to keep it in. PFK adds a second phosphate group onto it. So now we have our glucose 1,6-bisphosphate, or fructose 1,6-bisphosphate, sorry. So 2 molecules. That gives us, if you'll remember from our calculator from the last step, we now have our 2 ATP invested. So -2. Right? The next step is to split this molecule in half. Right? So beyond this, we now take a glucose 6 molecule. So we have a 6-carbon sugar here, and we split it now into 2 3-carbon sugars. Right? So now everything is essentially doubled, and this figure does a really good job showing that we split this right down the middle. 6-carbon now becomes 3-carbon sugars. So you can see I've also illustrated this in the figure. There's a little C. right? And in these steps, again, I won't make you memorize all of these single steps, but what we need to know is by the end of this, at the end of the day, we make an ATP twice. So here's an ATP on this 3-carbon sugar, and here's an ATP on this 3-carbon sugar, and we do that twice. So therefore, we get a total of 4 ATP total. With a net gain of 2 ATP. The last step is then to convert it to pyruvate. Pyruvate, again, is a 3-carbon sugar, and the important part of pyruvate is that it's then going to be used to pass on into the next step, which would be aerobic metabolism. Before we go too far and get into aerobic metabolism, which we'll do in the next, I want to bring us back to one other point in this diagram, and that is these NADHs. So this is in our net gains as well. We talked about them briefly in the last module, being great carriers. So let's actually define their roles here in metabolism as opposed to just talking about them in oxidation and reduction reactions. So NAD and FAD, as we'll see in the oxidative, are what we consider carrier molecules, right? They transport hydrogen ions and their associated electrons. Remember, we just kind of say those are the same in these situations to bioenergetic pathways for ATP production. In other words, they're going to take them into the mitochondria and pass them through the electron transport chain. I'm getting a little bit ahead of myself, but you'll know that is coming. So in glycolysis, we have NAD accepting an electron or a hydrogen atom in this case. Since it is accepting, that means it is gaining, right? So this is going from an oxidized to a reduced form, right? So it's going from an oxidized to a reduced form, and if we don't convert it back, if it's not restored, then ultimately glycolysis will come to a screeching halt, right? If we don't have this carrier molecule, then the process will completely stop. But don't worry, our body has the ability to do this. So how do we regenerate NAD in this situation? How do we get it from NADH back to NAD? First, if we have oxygen, this NADH is going to go into the mitochondria where it's going to give up its electrons. Those electrons are going to help form ATP and take part in aerobic metabolism. Again, aerobic metabolism, if we have enough, we'll recycle this, no problem. However, if we have insufficient oxygen, then pyruvate is going to accept hydrogen ions to form lactic acid or lactate, right? So here it is shown in this figure. So our glucose 3-phosphate, we reduce NAD into NADH. That NADH can then be recycled either oxygen-wise, or we can add it to pyruvate. So here's our NAD form. We can then reduce it, and the way that we do that is using an enzyme called lactate dehydrogenase. So what lactate essentially does is, you can see, it takes this H molecule and puts it then onto our pyruvate molecule and helps recycle NAD. NAD can then go back and work in this reaction right here. I have this again here in the next figure. So here, again, here's our glucose coming in. Here's our steps in glycolysis. I'll just draw it out for your sake, but we'll phosphorylate it first. This will trap it in the cell. Then we will phosphorylate it again using PFK. This breaks it into a 6-carbon sugar that has 2 phosphorylation steps. Here, then we break it into 2 3-carbons, and then continue to pass it down glycolysis. And then in this step, ultimately what we do is we take NAD and convert it to NADH. And then, assuming oxygen isn't present enough, we can take lactate dehydrogenase. Again, our same figure here. Look, pyruvate CH3CCOO. Right? Here we just replace, we take this hydrogen and move them. Right? That's good and bad because lactate accumulation can have a couple negative effects, right? It can increase tissue acidity because it is lactic acid as such. It's also associated with fatigue. We're going to spend a lot of time actually on this entire concept in lab. But it's also good for fuel for aerobic metabolism and used for a substrate for gluconeogenesis in the liver, which we'll talk about in the future, right? So the same enzyme, though, that can convert lactate has no problems taking lactate back to pyruvate. So ultimately, what it does is works in kind of a balance step, right? So we can produce pyruvate into lactate, and then if we need to, we can take lactate, shuttle it right back to pyruvate, and pyruvate can then proceed through aerobic metabolism.